Hello, thank you for the invitation. My name is Aymerick Eudes. I'm from the John Bioenergy Institute, which is one of the 4 bioenergy research centers in the US, supported by the Department of Energy. I worked in the feedstocks division at JBay, and today the title of my talk is Engineering Approaches for Diverting Lignin Biosynthesis towards the production of platform chemicals in plants. So this is to illustrate what we do at JBay. We produce bioproducts and biofuels using engineered microbes, which are grown using sugars and aromatics derived from lignocellulosic biomass. So in the feedstocks division at JBay, my role is to engineer bioenergy crops to facilitate this process. So my work is mainly focused on the lignin biosynthetic pathway, which is connected to the Shikimate pathway. Lignin is made of G unit and S units. These units are alcohol, G, derives from the polymerization of coniferol alcohol, and S from the polymerization of synapyl alcohol, and there's a little bit of H unit from coumaryl alcohol. So today I will show you the effect of expressing 2 bacterial enzymes in plants, QSUB and HCHL, and you will see that in addition to modifying lignin, the expression of these enzymes enable the overproduction of 2 aromatics in plants for hydroxybenzoic acid and protocatechate. So I will start with the QSUB approach. QSUB is an enzyme that converts 3-dehydroshikimate into protocatechate, which indirectly affect the pool of shikimate available for HCT, which is an important enzyme in the lignin pathway for production of G and S units. So Arabidopsis plants that contain the QSUB gene have a higher amount of protocatechate compared to wild type, less lignin, accumulation of H unit. This is spectra, 2D NMR spectra looking at the monomeric composition of lignin. And the biomass from this plant is easier to saccharify. So after various pretreatment and saccharification using cellulase, we get more sugars from this plant. So the QSUB approach, the QSUB gene was introduced into sorghum, which is a potential bioenergy crop. And these engineered plants also overproduce protocatechate compared to control. And they had lignin reductions as well. This was from plants grown in the greenhouse, and we also field tested these plants in Texas. We are in the process of analyzing the samples. What we know already is that the protocatechate production is maintained from these plants grown in the field. This year, we published the effect of having QSUB in switchgrass and other potential bioenergy crops. So QSUB switchgrass also has less lignin, which is particularly obvious on leaves. If you see here the lignin stain in red with the fluoglycinol, you see a reduction in the staining on some leaves of the QSUB transgenics. This plant also show higher sugar yield after saccharification. And we are also in the process of analyzing samples from the field. The QSUB switchgrass has been grown in 2 locations in California. We have several cuts, several harvests been growing for the past 3 years. In collaboration with Sean Mansfield at the University of British Columbia, we obtained QSUB poplar. And these trees overproduce protocatechate, up to 1% dry weight in the wood. They have less lignin and also an enrichment in H unit in their lignin. So at JBay, we process the wood from these poplar trees. We produced hydrolysates. We pre-treated the wood with ionic liquid and conducted some saccharification using cellulase and hemicellulases to obtain these hydrolysates, which contain aromatics and sugars. And you see that in the case of the QSUB hydrolysates, we have more aromatics, and this is mainly due to more protocatechate, but also more glucose and xylose. And these hydrolysates were inoculated with a yeast strain, a red yeast, Rhodosporidium, which is engineered to produce bisabolin, which is a potential jet fuel. So it's a biofuel. And you see that in the case of QSUB hydrolysates, this engineered yeast produce more bisabolin. So now I'm moving to the HCHL strategy. HCHL is another enzyme from bacteria that cleaves coumaroyl-CoA, the lignin precursor coumaroyl-CoA, into 4-hydroxybenzaldehyde. And Arabidopsis plants containing this enzyme overproduce 4-hydroxybenzaldehyde, but most of all overproduce 4-hydroxybenzoic acid. The aldehyde is converted into the acid inside the plant. These plants also show improvement of saccharification. But they didn't show less lignin compared to the wild-type plants. And instead, when we investigated the lignin monomeric composition using thioacidolysis, we noticed that in addition to the conventional units, G, S, and H, the HCHL plants accumulate rare C6C1 aromatics, alcohols, and aldehydes. And we think those are converted from 4-HBA. They derive from 4-HBA and incorporate lignin. And the incorporation of these rare aromatics in the cell wall has an effect on the lignin structure. So here you are looking at size exclusion chromatography profiles from lignin purified from HCHL transgenic plants and wild type. And you can see that in the case of the transgenics, there is an enrichment in the smaller lignin fragments. Instead, the large lignin fragments are reduced compared to wild type. So there's a reduction of the lignin size, a reduction of the degree of polymerization, which we think contribute to the improvement of saccharification. So the biomass from these transgenic plants was hydrolyzed using dilute acid. Here there was no cellulase treatment, so we end up with aromatics and xylose from hemicellulose in the hydrolysates. And these were used to grow Pseudomonas, which is a bacteria. And Pseudomonas was able to grow. It was actually able to consume the 4-HBA, which is in higher amount in the HCHL hydrolysates. And this Pseudomonas strain is engineered to produce another biofuel, a potential diesel fuel. And you see that in the case of the HCHL hydrolysate, the strain produced more higher titers of methyl ketones. In collaboration with this company, Futuragene, we developed some HCHL eucalyptus trees. At least one tree was expressing the HCHL gene very well and overproduced 4-HBA. The wood from these plants had improvement of saccharification, but again, no change in the lignin content, just a reduction in the polydispersity index, which is reflecting the size of the lignin. And in collaboration with Shinya Kajita at Tokyo, we received some poplar trees with the HCHL gene. At JB, we did the pretreatment with ionic liquid and the saccharification. These transgenic trees release more sugars, more glucose and xylose in the hydrolysates. And again, the conversion using the same red yeast I showed before resulted in more biofuel, more bisabolin production in the case of HCHL trees. And the last crop we transform with this gene is sorghum. We have two constructs for sorghum for two different promoters to drive HCHL expression. And you see that we obtain various lines with various expression of the transgene. So we are currently analyzing those plants. So I showed you how the QSUB gene can be introduced in plants, in crops, to reduce lignin, to introduce a low lignin trait in crops. And these plants also accumulate protocatecholates. So the next question we asked was, can we leverage this protocatecholate accumulation to produce something valuable inside the plant and stack the value-added co-product trait? And we decided to focus on muconic acid and 2-pyron-4,6-dicarboxylic acid, PDC. Muconic acid can be easily converted chemically to these compounds that are used for the manufacturing of several consumer polymers, such as nylon, foam, and plastics. And PDC is a functional monomer that can be used for the manufacturing of performance advantage materials, such as strong adhesives or more biodegradable plastics. So we wanted to test if in plants we can convert protocatechoate into muconic acid. And the two enzyme candidates that we tested for functional expression in plants are ROY and CAT-A. These two enzymes are also from bacteria. ROY is a decarboxylase for conversion of protocatechoate into catechol, and CAT-A is a catechol dioxygenase for cleaving the catechol ring and produce muconic acid. So these enzymes were expressed transiently in tobacco leaves. They were injected with agrobacterium in tobacco leaves, and successfully we were able to produce muconic acid in these leaves. We also included this enzyme from E. coli, ROG, to boost the overall shipment pathway. Recently, we built a large DNA construct for popular transformation. This construct contains the 4 genes, ROG, QSUB, ROY, CAT-A, under control of promoters, which we know are active in popular. So these are just very young transformants we obtain, and we hope they have less lignin. We'll analyze that soon because of the QSUB gene, and then produce muconic acid because of these ROY and CAT-A enzymes. For PDC, we used Arabidopsis, and we wanted to verify if protocatechoate can be converted into PDC using 3 genes, also from bacteria, from Novus Syngobium, Lig-A, Lig-B, and Lig-C. So ROG, QSUB, and Lig-ABC were assembled on a large construct using Arabidopsis promoters, and we obtained several independent lines. With this large construct, these lines have less lignin, which is expected because of the QSUB gene, and they also produce a high amount of PDC, up to 3% dry weight. These are the green bars, and you see that these plants, these Arabidopsis lines, still accumulate protocatechoates. So the pathway is not perfect. There's a bottleneck here that we are troubleshooting. And finally, we built a similar construct for sorghum transformation using these 5 genes under the control of constitutive sorghum promoters. So these are my conclusions. I show you that certain low lignin traits that, such as the one promoted by the QSUB gene, can be leveraged for synthesizing in planta some value-added co-product and doing some trace stacking. Muconic acid and PDC are 2 such bioproducts, and we believe that combining in planta synthesis of chemicals with the microbial synthesis can improve the overall titers of bioproducts. These are my acknowledgement. Xianwan and Yang are the two researchers working in my group on this project, Shinya, CJ, and Sean for the popular transformation, and for genetics for the sorghum transformation. Thank you. I'm happy to take questions if you have any.
Hi, everyone. My name is Luiz Henrique Catalani. I'm the present coordinator of Inova-USP, the University of São Paulo Innovation Center. I will start by thanking the organizing committee for inviting me and to give me a chance to introduce our Innovation Center. I believe the reasons for this invitation are twofold. First, because Inova-USP houses one of the most important initiatives in the area of biomass technologies development, the Synthetic and Systems Biology Center. And second, is to show how USP has innovated in its engagement in entrepreneurship activities by launching Inova-USP. The University of São Paulo has a very robust innovation system. You can check that out by watching Professor Marcos Martins from our innovation agency during this meeting. We are a very comprehensive university, very productive. We have been appointed many years in a row as the most entrepreneurial university in Brazil by the Association of Junior Companies of Brazil. Out of the first 10 unicorns of Brazil, 6 were originated at USP. The innovation ecosystem within USP is mostly under supervision of 2 different offices: the Dean of Research Office and the USP Innovation Agency. The agency is the final authority on innovation issues. It is our IP office, it's our TT office. It also looks at various regulatory issues within the university. About 3 years ago, a new initiative was launched, the University of São Paulo Innovation Center, Inova-USP, a new front end of the innovation at the University of São Paulo. So the idea was to create a specific enterprise devoted to foster collaboration with third parties in the area of innovation and applied sciences, private or public, universities or companies, national or international. We seek to create an internal and external reference, a beacon, a gate for incoming and outgoing actions of collaborative ideas and projects. The catch, or the lead idea, was to put a 5-story building with around 17,000 square meters to house the center and offer a fixed address for the innovation at the university. So, as we like to say at USP now, innovation comes with a zip code. At this very moment, we are starting Inova-USP spaces also in different campuses of USP, one in São Carlos and one in Ribeirão Preto. The idea is that we are going to be physically present in every campus of USP. Our strategic vision is to team up with the best people and labs within USP. We want to be able to offer easy access to our partners to the entire infrastructure and expertise available. We also focus on transparency and also on communication with all different actors inside and outside the university. Since we have the mandate to reach out to every institute, school, and faculty within the university, we are able to tackle more complex problems. We want to be perceived as distinct from monothematic centers, more like a very multidisciplinary research center. Our menu of university-industry interaction format is very broad. We are ready for a very complex type of interactions where a research center can be formed and where multiple projects and funding sources can be managed. Purely private sponsored R&D projects are the most valuable, but they are rare in Brazil. So we must seek within the sector funds fed by the clause of R&D and I investment. Consortia are important, especially for the low-budget companies. These are the most difficult, but we know how to do it, and we have a couple of examples in the house. Public money is scarce in Brazil, but we are privileged to have FAPESP in São Paulo State. Students of all levels are the main target through hackathons and a variety of activities. We prefer not to be a consulting agent, but we tend not to repel or refuse such needs from the partners. And finally, we plan to have a rich environment where we can attract investors of all types that can complete the chain with the startups. So far, these are the activities, projects, labs, or initiatives, as we like to call them, that are already in place at the Inova-USP. So let's have a close look at them. One of our most important missions is to capacitate our students for an innovative and entrepreneurial environment. Cosmos is our co-working place, where we intend to put our undergraduate and our graduate students in contact with this ecosystem. And we do that by offering competitions, hackathons, campus parties, demo days, startup weekends, and so forth. And then we have NIDUS, the Innovation Learning Environment. Its central point is to help shaping up startups by a strong tutoring program. NIDUS is a pre-acceleration program for graduate students, postdocs, and researchers already part of USP or wanting to be part of USP. It is responsible for the first residence program in innovation in this country. IRIS is run by a group of senior professors with the goal to steer more complex projects. They are called the Project Hunting Team. The strategy can be summed up in two scenarios. The first one, when they are challenged from outside, using ideation tools or design thinking, they reach out to USP experts and infrastructure to help solve their problem. Or when they are challenged from inside, again, using design approach, they look partners outside of the university, especially when specific funding opportunities are the goal. So Patium is the Laboratory of Games and Digital Solutions. It's a group involved in various digital technologies, like gaming technology, user experience, the factor of future, Industry 4.0, artificial intelligence, machine learning. So today they have already many projects in place. They have one center for artificial intelligence and machine learning that is sponsored by Banco Itaú. And also they have a center for artificial intelligence, which is sponsored by FAPESP IBM. So the next is SPPU, the Scientific Platform Pasteur USP. This is a long story. Since 2015, there is a tripartite agreement that was signed with Pasteur, the French Pasteur, USP, and Fiocruz, aiming to create an Institute Pasteur Brazil. So today, SPPU is the seeding facility to become this Institute Pasteur Brazil. They focus on the development of several infectious diseases, and I must say SPPU has been a key actor of the University of São Paulo on the efforts to help solve COVID pandemic problems in 2020 and 2021. So we also have planned a makerspace. It's called the USP Makerspace that we have planned as a first-class prototyping shop to be a reference within the university, so we can provide advanced prototype, support for design, inventing and building stuff for our partners. Although we are not a full educational unity in the sense of a faculty of a school, we do have in the house an undergraduate course. It is run under the responsibility of the undergraduate provost office. It's called Molecular Sciences course and is especially dedicated to the teaching of the scientific method. Students from any major can apply, from any major in the University of São Paulo can apply to CCM. So this final class, this group, is a blend of many different talents. They really have shown a great interest in the area of applied science, and they moved to Inova USP, and today they are fully integrated in the Inova USP environment. Okay, so last but not least, we have the Synthetic and Systems Biology Center, the S2B, the real reason why I'm here today. Other participants of this technical section are members of S2B. S2B is a center dedicated to the study of biology of various crops, focusing on the development of new technologies for the use of biomass. The goal of this research center is to promote collaborative and interdisciplinary projects in the area of biomass, including partnerships with companies. Their major focus today are the development of solutions towards reduction of CO2 emissions, to boost efficiency of the ethanol production chain from the field to the mills, and development of applications for the production of value-added molecules. This is a group that has been together since 2009. They are very tight-knit, very productive, and truly interdisciplinary. The group includes members from the schools, as you can see listed here, schools of engineering, pharmacy, biosciences, biomedical sciences, mathematics, chemistry, and agricultural sciences. And to work in conjunction with S2B, we have the CSBL, the Computational Systems Biology Lab. This is a bioinformatics group. They are devoted to solve biological problems through the development and the use of computational tools for the analysis of high-throughput data. As you can see in this cartoon, it shows some of their tools and databases. So far, this is the list of our partners. As you can see, we have a couple of public fundings from FAPESP, ABDI, and the Ministry of Health. But most of our partners are from private companies, very big international corporations, as you can see. IBM, Samsung, Huawei, Motorola, Isocetep, ArcelorMittal, Siemens, Schneider. So all of them very big companies. And we have big companies from Brazil as well. As you can see, Itaú, the big banker, Itaú is here. And this is just the beginning, and this list keeps growing. So thank you. With that, I close my talk, and I'll be happy to take questions from anyone at any format. Bye.
Hello everyone, I am Glaucia Souza, professor at the University of São Paulo. I'll talk today about our effort to develop genomic resources useful for crop engineering for energy and bioproducts, and also our plans for the Systems and Synthetic Biology Center established at the Inova USP, the S2B. Sugarcane is the world's highest tonnage crop, but its yields could go even higher. 10 years ago, we published this analysis on yield potential that concluded that the current yields of around 39 dry tons per hectare a year could be increased significantly with improvements on the environment, with new varieties altered for some key physiological traits, and with the use of biotechnology, towards what we believe possible over 120 dry tons of potential weight, or 380 fresh tons. This was for sugarcane. Now we have the energy cane that is producing an average of 46 dry tons per hectare of biomass. What I was talking about was based on sugarcane, where a sugarcane standard variety averages 37 dry tons per hectare. With the new varieties of energy cane, we see yield increases of 25%, fiber is increasing by 64%, and sugar is decreasing 44%. So energy cane with a higher fiber could be and low sugar will be very good for bioelectricity production. Those with increased fiber but still some sugar may be more suitable for integrated first generation and second generation ethanol production, for instance. But let's talk about molecules. What controls carbon partitioning and yields? What can I learn from the existing varieties and from their crosses? Commercial sugarcane is a cross between Saccharum officinarum, thick culms, high sucrose, low tillering ability, around 5 stalks per plant, and susceptible to stresses. These were crossed with Saccharum spontaneum, which have thin culms, low sucrose, high tillering ability, around 20 stalks per plant, and are tolerant to stresses. Said in another way, a modern sugarcane cultivar is a hybrid of officinarum with spontaneum, selected over many years to improve for sugar content, including back crosses to Saccharum officinarum. That led to a genome that has more than 10 copies of each gene in average, where most copies come from officinarum and only 10% come from spontaneum. What happens if I select for more spontaneum, more biomass instead of more sugar? To check that, we crossed spontaneum with modern sugarcane varieties. We did 4 different crossings, analyzed 210 genotypes, and selected the progenies with more tillering than with more biomass. On the right here, we have total biomass from the commercial cultivars SP803280 and RB867515, and next to it the total biomass of spontaneum, which were the parents of these processes. And look to the left here, we have the 2 of the progenies with over 250 tons per hectare, even higher than the energy canes that I have here as references. We next analyze the transcriptome of not only the high but also the low biomass progenies and also the parents. We use 3 tissues, 3 biological replicates, and 3 collection points of plants grown in the fields. And this is a heat map of the 200 most variable differentially expressed genes showing enriched metabolic pathways. We can see leaves, photosynthetic apparatus, drought sensing in leaves. In the upper internode, we see cell cycle activated, mitosis, growth taking place. And in mature internode, we see secondary metabolism, cellulose biosynthesis, lignin biosynthesis, fiber synthesis as expected. And this is an example of genes of interest for manipulation. We can see to the left here, we can see to the left here a gene that's probably derived from SP803280 in purple, that is more expressed in high biomass, and to the right a gene probably derived from spontaneum in blue, that is more expressed in high biomass. This is a way of identifying possible origins of the genes that may lead to improved yields and interesting targets for manipulation. The identification of these gene expression patterns is all right. It can aid in crop management, in crop engineering, but for overall understanding of crop development, we need to gather as much evidence on gene networks as we can before we even start altering genes. So the differential here for our analysis was that we have sequenced the genomes of SP803280 and spontaneum that were published in 2018 and 2019 in these articles of our Sugarcane genome references. We were able to address, with the integration of the multi-copy genome resolved with the transcriptomics, we were able to address also the main metabolic pathways altered during development of the plant. Using metabolomics, we added another layer of evidence for alterations of key pathways, and we were able to have an idea on how do climatic conditions alter SP803280 developments. And this, I am talking about drought, okay? So this is the best example I think we have so far, because we conducted so many different examples and we have very robust data that allow to put together gene networks associated to drought and how the plants and define how the plants are able to produce energy under the stress. We performed several drought experiments in greenhouse and field conditions in which 5 sugarcane varieties were subjected to periods without irrigation. We observed induction of most amino acid catabolism related transcripts that provide intermediates for the TCA cycle here in the middle. The higher induction is in blue, the decretion is in red, okay? The higher induction of tryptophan catabolism may be due to the fact that its degradation produces acetoacetate, which is here, and alanine, high energy compounds, which can be further catabolized to yield pyruvate and generate energy. By contrast, amino acid catabolism, the enzymes that do not provide precursors for the TCA cycle, for instance glycine, and those involved in the catabolism of amino acids for which it would be necessary too many steps of execution to synthesize the precursors of the TCA cycle, for instance methionine here, and glycine is here, they were down-regulated. Okay, and here we have the gene networks that pointed to these results and their respective nodes of regulation that can be targeted for engineering drought tolerance. We are now producing transgenics. At S2B, our Systems and Synthetic Biology Center at USP, we intend to try to bring to higher TRLs some of the technologies we are developing. We have taken some 10 years to bring some of the results to TRL5, such as with the sugarcane varieties added to the breeding program that I just talked about. They are part of the Ridesa breeding program now. We have some patents granted and some patents for drought tolerance license for genes of interest. But to really be able to control carbon partitioning, we have to also go towards the definition of what are the best promoter regions that could be used for gene expression regulators. We need to consider that sugarcane is a very complex plant. So going back to the beginning of my talk, I mentioned we have around 10 copies of each gene. One of the great outcomes of sequencing the genomes of both a commercial hybrid and the ancestor spontaneum is that we can now see which copy is responsible for the altered expression and how the promoter elements differ in each copy. I want to call your attention here to two genes involved in carbon metabolism. On top I have the SUSI, sucrose synthase gene, and on the bottom I have the PAU gene for lignin biosynthesis. Look at how many copies we have for these genes. Here's are the phylogenetic trees that we compose using the red dots are from SP803280 and the blue ones are from spontaneum. And we can see that the copies are expressed at different levels. This is expression profiles for all these genes. And look at all these elements. These are the promoter regions, how they differ. I want to focus on this one, for instance, just so you can have a peek. Look at this PAU that is more expressed in the internodes, how it is enriched in this transcription factor binding motif that we found. We are now conducting a high throughput analysis of sugarcane gene promoters to make synthetic sequences. We use two approaches for the in silico detection of TF binding sites. One is the de novo motif discovery, and two, the detection of transcription factor binding sites with known binding motifs. This is then followed by the assessment and selection of candidate motifs, which allow us to compute promoter architectures for the genes of interest, correlating the information to expression data and thus leading to a better understanding and characterization of the promoter sequences. Here is an example of the result of our promoter analysis workflow. We can see that some motifs seem to enhance expression, for instance the blue one. Others seem to decrease expression, and green, purple and orange motifs seem to be needed to maintain a high expression level. In their absence, we don't see, we see very low levels of expression. All of this is being integrated in the successive regulatory database. We have gene annotation, gene expression, public data, sequencing data, transgenic and metabolomics data integrated for a systems biology approach. I talked today mostly about increasing yields and genes associated to the energy cane, but our findings can help channel carbon partition to many compounds of interest. Remember that we make many components nowadays from sugarcane biomass, and we can make new ones with new properties that might be of interest. We are already investigating that. I want to thank, and by thanking all the hard work of my students and collaborators that have been working very hard out there in the sugarcane fields and setting up labs in the field for many years now, and also working very hard nowadays indoors. I also would like to end by thanking FAPESP and USP for funding this initiative. Thank you.
Hello and welcome to my talk. My name is Carlos Hotta, and today I'm going to talk about my work on sugarcane rhythms in the field. So most organisms live under a rhythmic environment. So you have light coming in on dawn, and then temperature rising during the day, and during the night you have darkness and then temperature going down. And those changes are mostly predictable. So one adaptation to live under an ever-changing world is to have an endogenous mechanism that keeps time, and we call this mechanism the circadian clock. So arrhythmic plants, they grow less than plants that have an intact circadian clock. So in these experiments using Arabidopsis, they overexpress a circadian clock gene, effectively stopping the clock during the morning. And those plants, they fix less carbon, they have a lower water use efficiency, and they are overall less productive than the wild type plants. So my lab wanted to know what happens when we look at the circadian clock of a very productive crop such as sugarcane. So in this experiment we have grown sugarcane under grown rooms, and we have put them under constant light and constant temperature conditions and harvested the plants every 4 hours for 2 days. And we have detected that sugarcane have a very high amplitude circadian clock, as you can see here in LHY, which is also a circadian clock gene, and TOC1. LHY always peaks right at dawn, while TOC1 always peaks at dusk, in the transition between light and darkness. And in this experiment we have shown that more than 30% of the sugarcane transcripts are regulated by the circadian clock. And this is much more than what we have seen in other plants using similar techniques. But we wanted to know what happens with sugarcane in the field. So we have gone to the field and taken plants every 2 hours for 26 hours, harvesting plants from right before dawn to right after dawn on the next day. And we have harvested plants that are 9 months old, so they are about 2 to 3 meters, and we harvested the leaves, we harvested the tip of the cone or the tip of the shoot, so it's meristematic tissue, and we harvest the 5th internode that's starting to accumulate sucrose. And we have seen that leaves in the field, they have a lot of transcripts that are rhythmic. Actually, more than 66%, more than 2/3 of their transcripts are rhythmic under those conditions. And that's more than in the previous experiments, because here we also have light and dark and hot and cold and other environmental rhythms aiding the circadian clock to control rhythmicity on the plant. But this in the leaves, and in the internodes we have much lower percentage of transcripts that are rhythmic, like 31% and 28.8%. But something that we found really curious is that whereas in plants under grown rooms, they have the LHY peak at the beginning of the day as expected and as measured in other plants, our plants had their peak at CT4 or 4 hours after dawn, and this was seen on the leaves in the internode 1 and in the internode 5. So what's going on? Remembering that those plants have 9-month-olds. So in order to check this, we also measured LHY rhythms in another batch that we harvested when the plants were small and only 4 months old. And those plants, they actually have their LHY peaking right in the beginning of the day. So plants, sugarcane plants that only have 4 months old, they are very small and they are like a knee high, whereas the plants that are 9 months old. As I said, they are very high. They have 2 meters to 3 meters high. So LHY peaked later, and we wanted to know if those other rhythms in the plants are also later in 9 months old sugarcane. So we did oligoarrays on the 4 months old sugarcane and the 4 months old sugarcane, and plants and like half of the transcripts on the 9 months old plants, they have a phase that's 2 to 6 hours later compared to the 4 months old plants. So it's not only LHY that's later, that peaks later, but almost half, more than half of the transcripts of the plants, of the 9 month old plants, they are peaking later, compared to the 4 months old sugarcane. And this also affects the metabolite rhythms in those plants. So here you can see the peak of the 4 months old plants and the relative peak of the 9 month old plants. So most of them are also happening later in 9 months old plants. So this is widespread in the plant. It's LHY, transcripts, and metabolites. So we hypothesized that what's making those peaks happening later is that 9 months old plants, they are being shaded by each other, and what's actually happening is that those plants are detecting dawn later. So that's why LHY, which is effectively a marker of dawn for the plants, is peaking much later in the day. To test that, we checked the plants in different sides of the field, because on the east side of the field, those plants are receiving sunlight right at dawn, and plants on the other side of the field are receiving light a bit later, actually receiving direct sunlight a bit later. And we have seen, and you can see here in a picture taken like a few minutes after dawn, what's happening in each side of the field. One side is all illuminated, in the other side is still shaded. And what we have seen is that LHY is peaking later on plants on the west side of the margin compared to the east side of the margin. And this is about 1.5 hour difference, almost 2 hours difference in the peak. And here in the graph it seems little, but imagine that you can sleep 2 hours more in the morning, or imagine having to wake up 2 hours earlier in the morning. It's quite a lot of difference, and it is a lot of difference for the plant as well. And so finally we tested those the rhythms of plants that are growing in one side of a wall and in the other side of the same wall. And this is a way to create an artificial barrier between the plants, because we wanted to show that this is just shading and not just shading avoidance response between the plants. And what we detected, and again this is a picture right after dawn, and you can see plants in one side of the wall being illuminated right at dawn, and the plants in the other side of the wall is still being shaded. And the wall was built here just 1 or 2 days before this experiment, and just this difference was enough to make LHY peak later, like 2 hours later, more than 2 hours later in this experiment. So we have, I think, compelling evidence to say that shading, and actually it's not just a light difference, but because we also have these impacts, temperature changes, these impacts, humidity changes. There are lots of environmental changes being impacted by shading. So what we say is that we are seeing difference in the microenvironment of the plants, and plants few centimeters apart that are under different microenvironments are actually behaving as if they are living in different time zones, because here plants are detecting dawn 2 hours later between each other. So our conclusions is that field microenvironments can change the plant circadian clock, and this highlights the importance of studying the circadian clock in the field as well as studying in the lab conditions, because in the field a lot of things are going on at the same time, and those can change the way the plant perceives the passage of the time in the natural conditions. And this have a lot of impacts in the plant, may have a lot of impacts in the plant physiology, and may impact the way for example flowering is triggered on those plants. So I would like to thank everybody that are watching my presentation. This work was mainly done by Luiz Dantas, which was a PhD student in my lab. Felipe Jesus and Maria Dourado also helped this experiment, and we also have a lot of collaboration with Camila Caldana, Glaucia Souza, Milton Nishiyama Jr., and Mona Lisa Sampaio. And this work was funded by Serra Pilheira Institute and the FAPESP under the bioenergy program. So thank you for watching my talk.
Hello everyone. The title of my seminar today is Lignin Metabolism, a Systems View. First of all, I would like to talk a little bit about what is lignin. Well, lignin is a cell wall phenolic polymer, a product of the phenylpropanoid pathway, a very complex and rigid polymer that is deposited in the secondary cell walls of specialized cell types, such as the xylem and fibers. Lignin is very important for plant growth and development because lignin performs basically 3 biological functions. First, lignin provides mechanical strength to supportive tissues, so it allows the plant to stand upright. Lignin also waterproofs the xylem cells, so it allows a very efficient transport of water and nutrients throughout the plant body. And also lignin is a barrier against herbivore and pathogens, so it protects the plant against these biological stresses. Lignin is not only important for plant growth and development, but also lignin has an economical implication because when we think about the scope of the bioeconomy, we aim to use plant biomass because the plant biomass is enriched in cell wall polysaccharides, and we want to degrade these cell wall polysaccharides into monomeric sugars, and then these sugars can be converted into a plethora of biocompounds, including biofuels and bioplastics in the biorefineries. This is that expectation. However, lignin is a bottleneck for this biomass processing. It hampers the access of hydrolytic enzymes and makes this process, the biomass process, not efficient. So one of the strategies in the field is actually the bioengineering of lignin in order to produce optimized biomass for biorefineries. And when we think about optimizing lignin metabolism, we have to first understand lignin metabolism. And this slide shows you an overview of several aspects of lignin metabolism, starting with the regulation that can occur in several levels, the transcriptional regulation, post-transcriptional regulation, and post-translational regulation, the biosynthesis, which is represented here in a very, very simplified form. Then the transport of the monomers from the cytoplasm, where they are produced, to the apoplasm, where they will be oxidized prior to their polymerization or their incorporation into the polymer. So all of those aspects can be a target for a lignin bioengineering. In our lab, we developed two model systems to study lignification in C4 grasses. C4 grasses is a very important group of plants for the bioeconomy, especially in Brazil, where we use sugarcane, which is, of course, is a C4 grass, for the production of sugar and bioethanol. So the two tools that I will talk a little bit today, that would be the xylogenic culture, an in vitro culture in which we can induce the differentiation of xylem cells, and xylem cells are enriched in lignin, and also the elongating internode, which is an internode that is still developing, so there it shows a gradient in secondary cell wall deposition. So we developed a xylem culture for sugarcane, in which we treated these cells with a combination of auxin and brassinosteroids, and we were able to induce the transdifferentiation of those suspension cells into tracheary elements, and those tracheary elements, as you can see here, they show a metaxylem-like secondary cell wall patterning, such as the pitted one, as you can see here, as this one, and the reticulate one, as you can see here in the top of this panel. Also, we followed these cultures for several weeks, and we measured lignin content, and you can see that in the induced cells, in the cultures that we induce the differentiation of these cell types, we see an increase in lignin deposition, which corroborates the formation of these xylem-like cells. Then we perform RNA-seq on those cultures. We clustered genes with similar expression patterns, and we could see, for example, that genes with this profile, which are induced in the cultures, in the transdifferentiation cultures, they are enriched in a biological process related to phenylpropanoids, flavonoids, flavones, secondary metabolism, reactive oxygen species, which means that they are inducing the expression of genes involved in lignification and programmed cell death. Whereas genes with this profile, where you see a downregulation of genes in the induced samples, we see a genes category of biological process related to cell division and cell cycle. In those cells, particularly when we evaluate genes involved in lignin metabolism, we see that most of the genes involved in the biosynthesis and also polymerization, they are strongly upregulated in the induced cells. So you can see here the induced cells are those represented by the gray bar in each catalytic step here. So you can see that for most of the gene families, the genes involved in lignin metabolism, lignin biosynthesis, are induced in the induced cells, corroborating actually the increase in lignin content through time, as we observed before. Then we generated co-expression networks from these transcriptomic data, because we want to identify genes that are co-expressed with non-lignin related genes, but that these genes might be also working in lignin metabolism. So we actually use this principle called guilt by association that states that co-expressed genes with unknown function with genes with the function in lignin biosynthesis might function in the same biological process. So by identifying genes with unknown function in the networks that are highly co-expressed with lignin-related genes, we are able to identify candidate genes that might also play a role in lignin metabolism in these plants. Of course, the next step, as I will show you in the end of this presentation, is to further confirm this function using functional characterization. Regarding the elongating internode of sorghum, we performed these experiments with both wild type and a mutant in the CAD gene, which is a biosynthetic gene in the monolignol biosynthetic pathway. We observed that in both genotypes we see a gradient in secondary cell wall deposition. You can see that in both genotypes we see an increase in lignin contents and also in xylose content, which represents here basically arabinoxylan, which is the major hemicellulose present in grass cell walls. And other monosaccharides representing here pectins, for instance, they decrease, which is of course that those results altogether, they suggest an increase in the gradient of secondary cell deposition, and this related to pectins decrease because grasses show very little pectins in secondary cell walls. So we confirm here that using the elongating internode we can see a gradient in secondary cell wall deposition. Then we performed phenolic profiling, and we clustered the compounds, the identified compounds in different profiles. So you can see here, this is the profile of the compounds through time from the base of the elongated internode to the top. So the base is less lignified, in the top is more lignified in both genotypes. So each of these clusters represent group of compounds displaying a similar accumulation pattern in this developmental set. And I would like to highlight two specific clusters. Cluster 3, this green one that shows compounds whose content increases through time only in the wild type, and those are basically oligolignals, which makes a lot of sense. So oligolignals are small lignin oligomers that are still not incorporated into the polymer. So the fact that we see much more of these lignin oligomers in the wild type makes sense, because the mutant is not able to produce normal amounts of the lignin units, of the canonical lignin units. So it corroborates our previous data. And also cluster number 1, which represents the compounds whose abundance increases only in the mutant, in this developmental set, only in the mutant, and most of them are derivatives of hydroxycinamates. So this pool of compounds represents how the phenylpropanoid pathway is rerouted upon CAD deficiency in sorghum. So here we can see a lot of other compounds here as well with the different profiles, including the decreasing in the content of chlorogenic acids in clusters 4 and 5, which corresponds it's an opposite trend to the accumulation of lignin in these internodes, which is chlorogenic acid has been suggested as a carbon pool for lignin deposition. So it makes a lot of sense as well. We also performed RNA-seq analysis in this developmental set as well. So we have the same aim to use guilt by association. So what you can see here as red dots, those are non-lignin related genes. So we are looking here for genes with unknown function that are highly co-expressed, because those genes, they might also play a function in lignin metabolism as well. So what we want with those two tools actually was not only to characterize those tools, but also to identify candidate genes for, of course, functional characterization. And we are starting these functional characterizations now. The two tools that I showed you before, they were developed for sugarcane in the case of xylogenic culture and sorghum in the case of elongating internode, but our functional studies have been performed in Setaria viridis, which is a model grass for C4 plants. You can see in the phylogeny here that Setaria is closely related to very important bioenergy crops such as maize, sorghum, sugarcane, and miscanthus and switchgrass. So it's a very valuable model plants for such functional characterizations. And we are also already looking for this data, so some of those genes that are co-expressed in our data sets before we identified their orthologous genes in Setaria. Some of them cluster here within these two subclades that are known lacases that performs a function in lignin polymerization in other species such as Arabidopsis and Brachypodium. Those lacases of Setaria viridis, they show, most of them show an expression pattern which is which is expected for a lignin related gene, or its expression is very high in lignification tissues, and we confirmed even here with in situ hybridization experiments that these genes are expressed in cell types that deposit high amounts of lignin. So now we are silencing these genes. We are still producing the transgenic lines, and in the future we will characterize those lines to confirm that those lines, they have a lignin-related phenotype. With this, I would like to thank you for your attention. This is my contact. Thank FAPESP for the funding, and thanks my group as well for the work. Thank you very much.